This is the 6th episode of Poke A Mon, where real science meets the Pokemon universe. I'm Kyukawobodo, but you can call me Professor Baobab. My expertise is in science communication. You can sign up to my weekly newsletter below to get access to a free ebook that connects your favorite Pokemon to real-world science. This ebook supersedes the guide I was offering before. Disclaimer: this podcast is in no way affiliated with the Pokemon Company, Game Freak, or Nintendo. I am using Pokemon as a pedagogical tool as it pertains to tangential learning. In other words, I tell stories using Pokemon to capture the attention of those interested in Pokemon while remaining integral to the presentation of scientific knowledge. Today, we'll be talking about cell division. Specifically, we'll focus on the different ways a cell can divide, called mitosis and meiosis. We'll also talk about the various phases of cell division. They're called prophase, metaphase, anaphase, telophase, and cytokinesis. Solosis is known as the Cell Pokémon. We'll use our newly acquired understanding to understand the physiology of Solosis' evolutionary line. Let's start off by talking about what is normally inside a cell. There are two kinds of cell in sexually reproducing multicellular organisms. These are called body cells or somatic cells and sex cells or gametes. These cells are similar with just a couple key differences. Somatic cells are found virtually everywhere in the body. They make up the vast majority of the 30 trillion cells that currently make you, you. I say currently because these cells can change over time in a process known as turnover. Every second, your body replaces about 4 million cells, equaling roughly 100 grams every day. Perhaps surprisingly, the vast majority of these cells are red blood. On the other hand, gametes are found only in the reproductive organs, i.e. the ovaries and testes, which are collectively known as gonads. The most important difference is that somatic cells contain a full set of genetic material, whereas gametes contain only half. This makes somatic cells diploid, which is Greek for twofold, and gametes haploid. In humans, each somatic cell contains 46 chromosomes. In egg and sperm cells, there are 23. By the end of this episode, we will understand why this is the case. Solosis is a cell Pokémon. Specifically, it's an animal cell. Animal cells contain a nucleus where DNA is housed, mitochondria, where energy units called ATP are made, cytoplasm, where various metabolic reactions take place, and a flexible cell membrane, which selects what goes in and what goes out. Solosis clearly contains a cell membrane and a cytoplasm. Its face could be a nucleus because it looks like it is about to split apart. However, when it evolves into Reuniclus, it suddenly appears like a fetus inside an amniotic sac. I'd also like to mention Deoxys before we begin our discussion on cell division, because Deoxys is known as the DNA Pokemon. What's the difference between genetic material, DNA, chromosomes, and genes? In multicellular organisms, also known as eukaryotes, genetic material is the DNA. DNA simply stands for deoxyribonucleic acid. You can surely see how deoxys comes from the D in DNA. In some single-celled or prokaryotic organisms, such as some viruses, the genetic material can be RNA, which can be thought of as a more primitive form of DNA. DNA is a long chain of nucleotides consisting of a sugar called deoxyribose, a phosphate group, and a nitrogenous base, namely adenine, thymine, cytosine, or guanine. Deoxys normal form clearly shows the famous double helix structure of DNA. DNA is coiled and condensed into chromosomes. Genes are specific segments of DNA that contain instructions for making a particular protein or RNA molecule. If you think of genetic material as a library, DNA are the books within the library. Chromosomes are the shelves where the books are organized, and genes are the chapters in the books where each chapter is about how to make something. I talked a bit about genes and genotype in the first episode called Evolution with Evie. Already, we have talked about somatic cells and gametes, diploid and haploid cells, and some features of the cell. For the remainder of this episode, let us place our attention on cell division. You might ask, how do cells divide and do what they are supposed to do at the same time? The answer is they don't really divide and perform their function at the same time. In fact, they mostly spend their time in a phase of 100% focus on their assigned task. This phase is called interphase. This seemingly occurs when Solosis is just about ready to evolve. This phase, surprisingly enough, is not mitosis. However, interphase is a necessary step before mitosis or meiosis can begin. This process is altogether known as the cell cycle. There are multiple cell cycle checkpoints along the way. These checkpoints are to minimize the chance of a mutation or worse. Initially, cells are doing nothing but what they were designed to do. Before the cell can proceed, it pauses to assess its environment. Is there enough energy? Are the conditions favorable? Has the DNA been damaged? If the cell isn't ready, it might not proceed and may be placed in gap 0 phase, which is like a maintenance phase. However, if the time is right, a cell will enter the gap 1 phase. During this phase, the cell begins to grow and produce new proteins and organelles. This segment is all about preparation. Enzymes essential for DNA replication are synthesized, while structural proteins are stockpiled. All of this sets the stage for what comes next and can take up to 100 times as long to complete as the mitosis phases. Yes, I said phases. In the games, Solosis normally evolves at level 32. However, if it decides to defer evolution, it can remain indefinitely in gap 0 phase. After a certain age, this is the fate for muscle cells, and to a large extent, nerve cells. People who work out don't increase the number of muscle in their bodies, but rather the size of their muscles. This is because muscle cells do not undergo mitosis, but they can grow larger, as evidenced by the physiques of bodybuilders. If all checks are passed, the cell moves into the synthesis phase, which is the stage of synthesizing DNA in interphase. Here, the cell focuses entirely on replicating its DNA, which takes just about twice as long to complete as mitosis. The chromosomes begin to unravel like a knitted jumper. Instead of yarn, chromosomes are made of tightly wound bundles of DNA called chromatin. Certain enzymes spring into action. Helicase unzips the double helix. DNA polymerase adds complementary nucleotides, i.e. adenine with thymine, guanine with cytosine, and ligase ensures DNA is linked correctly and there are no loose ends. By the end, the cell has 2 identical copies of its genome, packaged as 2 halves of a chromosome, known as sister chromatids. They are called sisters because they were born together from the same mother if you will, and are identical. At the same time, structures which will later help separate the sister chromatids, called centrosomes, are duplicated. Chromosomes work by migrating around the nucleus and pulling each sister chromatid to a different side of the cell using long fibers called mitotic spindles. They aren't doing anything yet though, as they need to wait for the final checkpoint. With its DNA now doubled, the cell enters gap 2, the final phase of interphase. This is a period of fine-tuning and takes the same length of time as the synthesis phase. The cell continues to grow, making final preparations for division. It produces the proteins required to build the spindle apparatus and further increases its stores of energy. Another checkpoint ensures everything is in order. Was the DNA copied correctly? Are there any errors to repair? If everything is fine, and miraculously it normally is, the cell is finally ready to transition into mitosis. If something did go wrong, and the defect can't be resolved, the cell may be programmed to kill itself in a process known as apoptosis. In essence, interphase is the intermediate phase between mitosis and normal cell life. The mitosis phases now begin. Even though there are a full 4 phases associated with it, as well as cytokinesis, it takes the shortest amount of time to complete. The 5 phases that happen in order are prophase, metaphase, anaphase, and telophase, followed by cytokinesis. To remember the order, you can use the mnemonic, Pokemon may attack trainers! Careful! Imagine that a Pokemon ranger standing at the entrance to Eternal Forest with a sign saying, Pokemon may attack trainers! Careful! The first 4 words help you to remember prophase, metaphase, anaphase, and telophase. The final word helps you remember cytokinesis. We won't go into the exact mechanisms by which the mitotic spindles are able to use metabolic energy to precisely separate the chromatids, and how the cell separates in the middle to form 2 identical daughter cells in a process of binary fission. As such, take the following as an overview of the 2 kinds of cell division in sexually reproducing organisms. The process of cell division by mitosis, but also meiosis as we will soon discover, is highly organized and depends on 2 major structures in the cell. The mitotic spindle, which is built from microtubules, and the contractile ring, which is composed of actin and myosin proteins, the same proteins that are found in muscles. These structures work together to ensure the cell divides accurately into 2 daughter cells. During mitosis, the mitotic spindle plays a critical role in dividing the replicated genetic material. Microtubules, which are dynamic fibers within the web of protein filaments in the cytoplasm, the so-called cytoskeleton, as well as specialized multiproteins, combine to create the mitotic spindle. Their job is to ensure that identical copies of the genome are distributed properly to each new cell. The process begins in Pokemon may attack trainers. Careful. Prophase. When the duplicated centrosomes, recall these are structures that will help move the sister chromatids, move to opposite sides of the cell, positioning themselves around the nucleus. This movement sets the stage for the spindles to form. Meanwhile, the membrane surrounding the nucleus breaks down. This breakdown is essential because it allows the microtubules from the spindle to attach to the chromosomes. Once attached, the microtubules begin to align the chromosomes in the middle of the cell in a process called congression. By metaphase, all the chromosomes now paired as sister chromatids or identical copies are lined up at the cell's equator. each sister chromatid in a pair is attached to opposite poles of the spindle, ensuring they will be pulled in opposite directions. Next, Pokemon may attack trainers. Careful. Anaphase. This phase is marked by two significant movements. First, the cohesion holding the sister chromatids together is broken. This requires energy, which is why the cells spend so much time growing and gathering energy in the form of ATP produced by the mitochondria. When each chromatid is pulled apart by the mitotic spindles, each chromatid moves toward opposite poles of the cell. This movement is known as anaphase A. Simultaneously, the spindles themselves elongate to move the centrosomes further apart, stretching the cell. This is called anaphase B. These combined movements ensure that each daughter cell will receive a full set of chromosomes. During anaphase, the mitotic spindle also sends a signal to the outer layer of the cell. This signal determines the position and orientation of the contractile ring. The contractile ring, as the name suggests, is a ring-shaped structure which contracts to pinch the cell in two and is the main driver of the next and final step of the cell cycle, cytokinesis. Cytokinesis begins as the contractile ring starts to contract, forming a groove that deepens as the ring tightens, or a cleavage furrow. This contraction effectively divides the cytoplasm between the two daughter cells. Meanwhile, the nuclear envelopes start to reform around the separated chromatids, which begin to take on their normal structure within the nuclei of the two new cells. Finally, the furrow pinches completely shut, sealing off the two cells from one another. At this point, the two daughter cells are fully separated, each with its own complete set of chromosomes and cytoplasmic contents. Hopefully, the cells are exact clones. Earlier, I claimed that the vast majority of cells are these somatic cells that only reproduce by mitosis. We have now seen how mitosis works to create exact replicas of cells. So, how come we aren't all just clones of one another? In fact, how come every cell in our body isn't just the same cell? This is where meiosis comes in. Now, meiosis is very similar to mitosis in operation, so I won't bore you by repeating the same things. Just know that meiosis also starts with interphase before moving on to prophase, metaphase, anaphase, telophase, and cytokinesis. The difference is that in meiosis, prophase, metaphase, anaphase, telophase, and cytokinesis happen twice in succession. This does not mean meiosis is the same as mitosis happening twice. Somatic cells can also undergo mitosis twice, but that doesn't mean they undergo meiosis. Meiosis only happens to make gametes. The germline are cells in the body that produce the haploid cells, which we call gametes. They themselves are diploid and eventually produce diploid cells when a male and female gamete successfully form a fertilized cell called a zygote. This zygote is a cluster of undifferentiated stem cells, which basically means they could become any type of cell, kind of like how Ditto can change to any form, except this transformation is permanent. Curiously, when Solosis has finally evolved, perhaps occurring when the cleavage furrow has finally pinched through to create twice the number of DNA, it turns into Duosion, the Mitosis Pokémon. Despite being called the Mitosis Pokémon, though, Duosion does not look drastically different from Solosis. You might have expected something like Magnemite evolving into Magneton, where they literally gather identical copies upon evolution. But no! Instead, Duosion has a larger body that makes it look like Cow from Sonic Adventure 2. A single germ cell from the germline contains 23 pairs of chromosomes in humans because it's a diploid cell. When meiosis occurs, a series of 10 phases occur. These are prophase 1, metaphase 1, anaphase 1, telophase 1, and cytokinesis 1, followed by prophase 2, metaphase 2, anaphase 2, telophase 2, and cytokinesis 2. It has been theorized that meiosis evolved from mitosis, hence why it literally seems like the prequel to the movie called Mitosis. Instead of saying prophase 1, metaphase 1, etc., I will collectively call them meiosis 1 and meiosis 2. As such, we have 2 cell divisions during meiosis, twice the room for error. No wonder cells evolved to be so stringent when it comes to quality control. Any defect in the daughter cells of the germline can result in miscarriage or disability in the offspring. Diploid cells contain 23 matching sets of chromosomes in humans. In other species, it will be different. These chromosomes, like pairs of shoes, are not identical but match because their genes are similar. Each pair is therefore referred to as homologous. For the germ cells about to undergo meiosis, an identical sister chromatid is attached to each chromosome, so the cell has the same number of chromosomes, but with twice the number of chromatids during interphase. Just before the meiosis 1, chromatids of different chromosomes cross over, literally, sections of their genes. This process is part of what is known as genetic recombination. This may ensure genetic diversity in the gene pool. Genetic diversity is important for a number of reasons. First, if both your parents suffered from a genetic disorder, you may not necessarily inherit the disorder. Furthermore, the success of a species is normally linked to its genetic diversity. We saw what happens when there is not much diversity, when a single disease wiped out a huge chunk of the population of cultivated bananas. Finally, genetic diversity is also the driving factor for evolution. Without genetic diversity, we would not have the diversity of wildlife we see today. Complex life wouldn't have evolved. So, after crossing over, the cell divides once in meiosis 1 to form 2 unidentical daughter cells with 23 chromosomes. Then, the sister chromatids are split to form 4 unique daughter cells with 23 chromosomes again, but with half as many chromatids. Recall that a chromatid is effectively half of a chromosome in diploid cells. It is these cells that we refer to as gametes. If the germline was near the ovaries, the daughter cells would be eggs. If it was near the testes, the daughter cells would be sperm. Most of the daughter cells that form near the ovaries do not form into full eggs and are reabsorbed by the body. When the sperm and egg fuse in the process of fertilization, the 2 cells combine all their genetic material into one cell called a zygote. This zygote is diploid because the 23 chromosomes from each gamete pairs up to form 23 pairs of chromosomes. To clarify, each chromosome is bound together by something called a centromere, essentially holding it in place and assisting in the motility of the chromosome. Finally, the zygote divides by mitosis, as only germ cells can divide by meiosis, to form cells that will eventually differentiate to become cells for the bones, the lungs, the brain, etc. Reuniclus may be showing what happens when a zygote develops into a fetus. A fetus is surrounded by amniotic fluid from the womb. However, the spots surrounding Reuniclus may still be organelle, so it might be painting a picture of the results of mitosis and meiosis simultaneously. If you've come this far into the episode, please consider liking this on Spotify, Apple Podcasts, etc. This will help me tremendously and cost nothing but a few moments of your time. We talked about cell division, mitosis and meiosis, the cell cycle, and the phases: interphase, prophase, metaphase, anaphase, telophase, and cytokinesis. We also talked about somatic cells and gametes. Somatic cells are diploid, while gametes are haploid. You can find the resources I consulted while researching for this episode in the show notes. Isn't the power of science simply amazing?